which is coming from the field. These are all very big challenges, and Dr. P. Usha Sarma has long experience in working on Aspergilli, and I am very much hopeful that with Madam's insight, all the scientists who have assembled here, they will be greatly benefited. And Aspergillus is a concern not only for maize, but groundnut, then chili, and the other field crops as well as horticultural crops are also getting affected with Aspergillus. So I hope that Madam's lecture on this particular area will trigger us to think differently, and we'll be able to come out with a very detailed research project, that how we can address it, and all the scientific community will be benefited with her talk, and the kind of direction we'll be getting. I think that will really help us. I'm very much thankful to Dr. Dubey, our Assistant Director General Plant Protection, that it was he actually who talked to me about Madam Usha Sarma, and then I contacted her. So Dr. Dubey, I am very much thankful that I know that you had very busy schedule. You will have to prepare some important document for parliamentary committee. So I welcome you. I welcome all the scientists from maize family as well as other crops like groundnut and other horticultural crops, and all other scientists who have joined this event, and I'm very much sure that they will be very greatly benefited. So before I request our chairman to give his opening remarks, I request our principal investigator, plant protection, and our senior most principal scientist, Dr. J.C. Shekhar, to introduce our speaker. Dr. Shekhar, please.
Yes, sir. Thank you very much, sir. Good morning, friends. Honorable Dr. S.S. Dubey, sir, ADGPP, and esteemed speaker, Dr. P. Usha Sarma, Madam, our beloved director, Sujay Rakshit, and friends from AICRP and IIM family, other ICAR institutes who have joined in this program. I am honored to introduce Madam P. Usha Sarma in this program. Madam holds PhD degree in biochemistry for Usmania University on structure function relationship of proteins and enzymes. She has more than 40 years of basic and applied research experience using multidisciplinary approaches in the areas of Aspergilli, host pathogen interactions, development of immuno and molecular diagnostic therapeutics. As the leader of molecular biochemistry and immunology section at GIB, she trained 18 PhD and 10 MD scholars and has to her credit more than 160 publications, including chapters in books, international patents, and importantly, numerous CSCs, SNPs, protein and polypeptide sequences of Aspergilli submitted to Gene Bank or Protein Bank. Dr. Sharma retired as a director grade scientist from the Institute of Genomics and Integrative Biology, CSIR Delhi 2004. She continued with the pursuit of her research interests as CSIR Emeritus scientist and later as an honorary scientist at the Division of Plant Pathology of the ICAR-Indian Agriculture Research Institute, New Delhi, working on aflatoxin biosynthetic pathway in Aspergilli. And Madam also developed WHO recommended clinical diagnostic kits and transferred the technology to industry. She developed an immunodiagnostic kit for aspergillosis and offered diagnostic services to society for several years at IGIBI for aspergillosis. She also provided consulting services to biotech industries both in India and abroad. She served as PI coordinator of several research projects sponsored by CSIR, DST, DBT, ICMR. And Madam had several national, international collaboration with scientists, professors of PMA institutions in India and around the world. She was visiting professor to DNA Technologies Division of National Institute of Science Technology, NIST, and Center for Advanced Research in Biotechnology, USA, Medical Research Center, Oxford, VA Medical Center, Wisconsin, University of Campus in France. She has delivered several invited lectures both in India, USA, UK, France, Australia, Japan, and Norway. And Madam is a fellow of several scientific societies such as Indian College of Allergy and Immunology, Association of Clinical Biochemists of India. She also was president of ACBA and organized several international scientific conferences. Throughout her career, she has received several awards. This include the Atomic Energy Merit Scholarship, DBT Senior Overseas Research Fellowship, INSA Royal Society Fellowship, and NIST a Guest Scientist Award and Regional Service Award by the APCC. I am very honored to introduce Madam to today's program. And now, Sir, Dr. I hand over the program to Dr. S.C. Dubey, Sir, Chairman of today's program. Thank you very much.
Thank you, Dr. Shekhar. Respected Dr. Sarma, Dr. Surya Rachit, Director, IIMR, fellow scientists and others, those who are attending this lecture. As Dr. Sarma has been introduced nicely, I would like to mention here and also request to Dr. Sarma, actually this lecture series is updating our knowledge in respect to the Aspergilli and further to utilize this skill to develop a project to manage the Aspergilli crop in general and particularly in the maize. As it has been already stated, I knew Dr. Sarma from last 20 years when she's working in the IARI Division of Plant Pathology. Little bit I got some technical backup from him also, even because Aspergilli, I never handled the Aspergilli, but the toxins producing and non-toxins producing at that time, emeritus scientist, Madam was working, and very nicely she has invented the pathway and the genes responsible for the toxins producing Aspergilli and non-toxins producing Aspergilli. I remember this information I used to present in the one institute mega program on sanitary and phytosanitary, the institutional project. So, Madam, I would like to request you, we wants to know the detection procedures for the toxins producing and non-toxins producing, because this will give the base knowledge to formulate a project to address the challenges into the Aspergilli. And detection not only the toxin producing, but in the product, if any final product, suppose the flour is there or any cake is there, how we can detect whether the aflatoxins level, the lot of procedures are there, but if the contamination level is so low, how we can detect and which method is much more efficient, and if we wants to refine this detection technology, what would be our approach further. Similarly, in the management aspect, because most of the things is required to ultimately has to be managed, and management in the field also to minimize the infestation of Aspergilli, and similarly in the post-harvest management, what would be our ideal approach. A lot of cultural practices, avoiding the moisture, avoiding the contact, and all these things are there, but even then Aspergilli is so much commonly present in the environments, they are getting our produce is getting contaminated. So in this way also we need a certain directions in which direction we have to in which way we have go forward to devise a very efficient and effective management strategy to reduce the aflatoxins producing contaminants, and leads to the rid of the aflatoxins production into the produce. And normally, as I understand, if suppose non-toxins producing Aspergilli, even that is also very dangerous to the agriculture, especially in the fruits. They are colonizing, have the ability to produce the certain enzymes, cell wall degrading enzymes, pectin degrading enzymes, and these enzymes, this degradation leads to the infection of the secondary bacteria or other fungus like the penicillium and others. Lot of fungus infection is there. So this has also to be addressed in which way this can be managed. These are the few points why I am mentioning that while delivering the talks, definitely the madam will touch upon all these points, and if any clarification is required from audience, definitely they have to interact with madam. This is not a just like a lecture given by the madam and you have to heard and we forget it. Definitely especially the person, those who are planning to make a project, they must interact very fruitfully with the madam to get the idea how, in which shape we have to proceed further to make a very good and effective program, not only for detection but the management of Aspergilli. With these few words, once again I am welcoming madam. For several times I heard madam's lecture, but definitely every time it is new and very informative for all of us. So on behalf of the organizers and my own behalf, I thanks to the madam for accepting our invitation. As Dr. Rachit has already told that when he was discussing with me, somebody has to give a talk on the Aspergilli and we want some technical guidance. Immediately, I remember you, but unfortunately in this phone, I don't have your number. Then I left to Dr. Rachit and told him you contact someone in the plant pathology and definitely you get the number of madam, and no one is better than the madam to deliver on the, in India at least, on the, because he is the globally well known for the Aspergilli. Thank you, madam, for accepting our invitation. And now I request madam to deliver your talk.
Thank you, sir, for your introductory remarks. So, and again, I thank you for introducing us to madam, and I am pretty sure that we will be very much benefited. Now, before madam starts her presentation, I request all of you to keep your video on for 5-10 minutes so that we can grab the screenshots. Madam, now I request you start your presentation, please.
Okay. Thank you very much, Dr. Rakshit. First of all, you invited me to talk about some of our work and experiences and also relevance to the problems which you are facing for with the aflatoxins. And secondly, thank you very much, Dr. Dubey, for your so many kind words you have said. I hope I'll deserve all of them, what you say, at least some of them. And then I would start my talk on Aspergilli, especially in this 75 years of freedom. And then especially this year is a celebration of 75 years. I feel this is a lot of scientific advances have happened. And then the scientific advances which happened even in the understanding and dealing with the Aspergilli is quite a bit. A lot of change was there. So first, that is what I just want to thank all of these people. Then the next one. Just a second. That. Yeah. See, this is looking very appropriate in science and in many fields of any knowledge. That definitely I want to read this for all of us. The more I live, the more I learn. The more I learn, the more I realize, the less I know. So these are the words of Michael Legrand, and very appropriate for many subjects and many things. And then this is how it gives the thing. So before I really go for actual work-wise things, definitely I like to acknowledge my really sincere thanks and gratefulness to my clinical collaborators who were Dr. Chetty from AIIMS and Dr. Shah from VPCI, KJR Murthy from Havir Hospital, Professor Denning. And when I was working at Center for Biochemicals, which is located in VP Chest Institute, we had discussions, several times discussions with the clinical people, and finding there is a big problem in the diagnosis of aspergillosis in pediatric patients as well as in the adults. So there is a need for diagnostic detection methods for Aspergilli in the humans. So this made me to rush to the IARI. That time, Dr. Sarabhai was there as the ITCC in charge. I collected an isolate from there. I started all my work later on with that. And several other collaborations are also there for molecular biology, proteomics. So I deal that in the appropriate time. Why I want to deal something about the humans and Aspergillus is because it is also relevant for our human health, which is a common factor for all of us to know. So that is why the thing. And as Dr. Dubey explained to you that I joined as an emeritus scientist in ICAR-IARI, and that time Dr. Nagarajan was there, and Professor Anupam Verma and D.V. Singh and all subsequent heads of the departments have been very kind to me. I continued as, and that interest has my change of interest from my health to the polypeptide synthesis and aflatoxins in the relevant for the agricultural field. And also I must say that why I have taken aflatoxinase during my retirement time, 2003, in that last year period, I could get a cDNA clone of polyketide synthase unexpectedly when we were trying to look for drug targets and other things, many other new allergens, etc. I have come across polyketide synthase. Immediately I realized this is highly relevant for the aflatoxin production and relevant to the agricultural crops. That is the reason why I have gone. I have taken that as a topic for my post-retirement work. And we have here Aspergillus flavus and parasitics, which are known to produce mycotoxins, such as aflatoxins, ochratoxins, cyclopiazinic acid, which are all, and many other toxic small compounds are produced by these species. And Aspergillus species also produce a lot of medically and pharmaceutically, commercially, and important bioactive molecules relevant for biotechnological utilization. It is our efforts to see what we can retrieve from these organisms for the benefit of the humans is a big task here. And when it comes to the humans, it is glycoproteins, allergens, enzymes, multifunctional enzyme toxins. They're all immunologically active molecules. When the fungus invades the humans or we inhale the spores, these are the molecules which are creating a havoc in our health conditions. So flavus is particularly interesting for everyone because it is not only human pathogen and it is isolated many times. It is also a plant pathogen, as Dr. Dubey already explained. These pathogens affect very badly on the peanut, corn, and many spices and oils and dry nuts. So the economic impact is very high, and we are the losers if we don't do in a speedy manner to contain some of these aspects on the contaminations. Now, when it comes to the human health, we develop antibodies when we inhale the spores, and continuously we are affecting our immune system is not very powerful, and we are having susceptibility for allergies and asthma. Then we develop antibodies to these Aspergilli. So, and these antibodies are not for the protection, but they are for the pathogenesis. So we are constantly exposed, and we need to have a methodology to detect these antibodies in the patients to contain the disease, which is going to be very harmful and leads to the mortality and morbidity of the humans. And here is a WHO report, actually, they have bulletin which has come in 2011. So number of cases of deaths of tuberculosis are due to the undiagnosed aspergillosis because the symptoms are very similar. So both the lung x-rays show a similar fleeting shadows and patterns. And in both the cases, fever will be there, cough will be there, chest x-ray is the same. And anti-tuberculosis treatment, tuberculosis patients, they recover, whereas the people who are having the aspergillus contamination in tuberculosis, they will not. They become the anti-ATT resistant also many times. So the antibodies in such patients is important to go through. Now, pulmonary, as I said already, pulmonary aspergillosis is mistaken as a tuberculosis. And then the progressive disease is resulting in many clinical damage and finally mortality. Several reports we have got in this paper from our own country where they have demonstrated the interference of aspergillus in tuberculosis. And this is the great bulletin 2011. It said that developing countries and in Asia Pacific region, it's a big burden of chronic pulmonary aspergillosis as a sequel to the pulmonary tuberculosis, besides bronchial asthma and also allergic bronchopulmonary aspergillosis patients. The target of the population who require such a type of diagnostic test is bronchial asthma people, allergic bronchopulmonary aspergillosis, tuberculosis patients. And of recent, I wanted to present this because in connection with the corona and COVID-19 patients, there are reports in 2021 saying around 90 to 33% in certain studies, they are undetected aspergillus infection. We know mucormycosis and then mycosis is happening, but here and they are treated with the antifungal drugs. So this is an important point that it should be detected even in the these patients early for better situation. So I just want to be developed a diagnostic kit based on the peptide for human ABPA and human aspergillosis. And just quickly, I just want to go, a lot of researches have happened over a period of several years. The initial isolate what I took from ITCC from PUSA Institute, then I tried to make a culture filtered proteins and develop an ELISA fractionated proteins, and we purified proteins and cloned and sequenced several new allergens and proteases from Aspergillus fumigatus and characterized them. And when we did the sequencing of an 18 amino acid protein, we have done an epitope mapping for that. This epitope mapping resulted in identifying a particular 12 amino acid sequence from the N-terminal region. And this peptide has been extremely relevant for commercially viable diagnostic kit for aspergillosis. This is the peptide after bopping. It has a B and T cell epitope from the N-terminal region, and it is cloned, sequenced, and characterized by us. And here is then, we try to, before my retirement, I have carried out the studies to come up to certain level with the help of all of our students who are really dedicated and they have contributed a lot. And then it is continued by Dr. Taruna Madan, one of my very dedicated and intelligent student. And she joined as a scientist at ICMR Institute, but still continued to make this peptide for a commercial utilization and public utility for the human utility. So she got a license. We have a DBT project has been given to us. So test manufacturing license was obtained for the development of IgG and IgE at GMP certified facility with SPAN diagnostics. To make a science to a relevant translatable thing and then make a commercially viable product demonstration is not a very easy thing. But then with the help of, I think, collaborators and appropriate funding, this has been able to reach a stage where it can go to commercialization. This is the GMP facility, manufacturing facilities. And these are just to show the kits which have been developed by us, and both for specific IgG and IgE antibodies, which is highly relevant even today for the corona situation. And quickly, I am just showing the data where it is relevant to screen the tuberculosis patients and smear negative and ATT drug resistant people, and several initial diagnostic dilemma situations can be handled by this kit. The results were submitted to ICMR with great sensitivity and specificity of this kit, and distributed to several parts of the country in the hospitals who validated. And the complete document has been submitted to ICMR, which approved finally to see the screening of ATT non-responders for aspergillosis in the ICMR's National TB Surveillance Program. So quickly, I say that these are all the inventors from IR and NIRRH. Especially main person is Taruna Madan, and Priyanka was responsible for epitope, and PGH Chandigarh, and we had a lot of collaborations, and these people are all, they have contributed to see the product to this level. Now, quickly, I just say host pathogen interactions are relevant for humans as well as the plant system. Here with the human system, when we try to understand what is happening for this Aspergillus entering and what is it happened to the human, then we identified, we knew this work has some about lung surfactant protein. People have already done the work which was relevant, but they haven't, nobody has done on the Aspergilli and fungal part of it. So luckily we got a great collaborator, Professor K.B.M. Reed from the MRC Oxford, who was handling the surfactant proteins. These surfactant proteins are very complex. They are present in the lung type 2 Clara cells, and they're also present in other cells. The main thing is they contribute to the lung innate immunity. So they contribute to the antimicrobial activity. So this protein was being studied by Reed's group with the help of Dr. Uday, who was a main collaborator for us even subsequently. And this protein has got a much relevance everywhere now. Is a multimeric protein. It has got a N-terminal region, collagen region, and carbohydrate recognition domain. So this carbohydrate recognition domain is going to bind to the surface of the glycoprotein of the microbe, and it leads to the pathogen phagocytosis, lysis, and then macrophage killing and maintaining the immune regulation. This is how this protein is highly relevant for our innate immunity for the lung. Our studies, this was a great slide to show how the surfactant protein and the conidia and the spores could react. We could demonstrate this in vitro and show the phagocytosis and breakdown of that in a dose response manner. So the surfactant protein has got a therapeutic value here for the human situation. And further, this work also led to the development. We have to demonstrate with the animal model. And then this is an invasive model, and then control groups we have done. And here also it is clearly showing that the hyphal density has decreased with the treatment of SPD. These are all the published work with the high impact journals. And here the eosinophilia is another complication of the lung fungal allergies. So this also showed that clearly eosinophilia is decreased. So a recombinant SPD was prepared by Dr. Uday. So this was to be inferences that SPD and MBL, mannose binding lectin, they showed therapeutic potential in the murine models of invasive aspergillosis, and only SPD showed therapeutic potential in the murine model of ABPA and G knockout mice. So extensive work has been done to show the therapeutic application of this particular surfactant. And what is its status today is that people have done a lot of work on the surfactant protein and the spike protein of the coronavirus. And here the spike protein and coronavirus docking experiments clearly demonstrated as hypothesis, recombinant human SPD can competitively inhibit interaction of spike protein SARS-CoV-2, and it can be used for the further purpose of exploring for a adjunct therapy in the humans. So that is the later. And here is again a novel adjunct therapy for inhalation of formulation of recombinant SPD was being worked out, and Dr. Taruna is making a product with this in an inhalation product. And then let us see how it goes. They are working on the animal models, and finally the product is expected to help our people. Now the final collaborators are here, Dr. Reed and Uday Kishore and MBL for Stephen Thiel. Without acknowledging them, the work will not be complete. We have got certain molecules from them, and this is the thing. Now the final part of the thing, which we have worked for last several years, is the Aspergillus species and their compounds first. So flavors is, as I said, it's already human pathogen and plant pathogen. It has glycoproteins, proteins, aflatoxin B1, G1, G2, M1, and aflatrem, cyclopyosinic acid, sterigmatocystine, and versicolorine. So these are all the things which are produced by flavors. Industrially relevant compound urate oxidase is also produced by flavors. Parasiticus is also relevant for our things on the agriculture. That is, they produce B1, G1, G2, M1. And Niger has kozic acid, and fumigators, as I already talked about that. And Vorize is relevant for biotechnological approach, and then food industry, lipases and celluloses. Ochraceous, ochratoxin is produced by that. And these are certain things which are produced by that. Here, the genome sequencing information is highly relevant for tackling the problem of Aspergilli in different ways, for the contamination or for identifying new compounds, and for every aspect of the problems. Here, the genome information of Aspergilli is highly relevant, and general information is here. There is so much to know and understand which are not looked into. That is the scope of the work for both biotechnology, for agricultural things, and pharmaceutical and drug things also. Important polyketoids here are lovastatin, is a anti-hypercholesteremic reagent, which was first isolated from the terriers. And then, as we know, that aflatoxin, cyclopyrazinic acid, stigmatocysteine are relevant for our agricultural crops for the things. And the biosynthetic pathway, why as per polyketoid biosynthetic pathway is relevant for aflatoxins is it is one of the pathways, secondary metabolic pathway, because of through which they produce aflatoxins. So we have taken here mainly a melanin pathway, which is also a important compound from the fumigators, and aflatoxin pathway for the flavors and parasiticus, and lovastatin pathway for the Aspergillus terriers. When we see the starting point, starting reaction is acetyl-CoA and melanyl-CoA. These are the two important molecules which react together to synthesize a pentaketoid. And then in case of Aspergillus flavus, it is a hexanoyl one. And in case of lovastatin pathway, it is also a hexanoyl X. The important thing is, though it polyketoid synthase biosynthetic pathway exists in all Aspergilli do not produce the toxins. And in some of the Aspergilli, this pathway is cryptic. It is not expressed. It is not expressed, because of which the particular species will not produce any toxins. So the difference, why it is there is a big question. It is present, it is cryptic. So can we make physiological conditions to stop the toxin production, and also make the other species which are not producing to make them produce under certain environmental conditions? So the biosynthetic study of biosynthetic pathway is a very important thing, and that is a basic science which requires to study, especially in relevant species. Now going to the polyketoid synthase is the major enzyme which is relevant for the synthesis of the aflatoxins. And this polyketoid synthase is a huge, big enzyme, and the 1,000 amino acids are there. And the gene architecture has got several multi-domain enzyme. This one is a multi-domain enzyme. And the main domains are the ketosynthase and acyl transferase, dehydrogenase, acyl carrier protein, and certain condensation reactions take place. So, and then some cyclization reactions take place depending on the product which it has to produce. So the key enzyme PKS plays a major role by having its own way of dealing with the organism to produce the compounds. So what we have done is we try to study a bit of it on the ketosynthase, polyketoid synthase, and the ketosynthase domain, which is essential for studying and initiating and continuing the product formation. So a lot of bioinformatic work has been carried out by Dr. Preeti with the help of bioinformatic division at with the Yoginder Singh and his group. And here we have taken the 3 partially reduced by doing the modeling of the ketosynthase domain. They identified certain active sites and also finding out differences between the species which are relevant for the type of product each produces, each toxin produce, or each statin, or whatever the thing. So then the next thing what has been done here is that the sequences were aligned and try to see there is what is the significance of that. We found certain conserved regions in these polyketoid synthesis sequences. So we noticed these particular ones and tried to develop some PCR primers based on the conserved regions and unique motifs which are there, and try to make a detection method for Aspergillus species based on a conserved region of ketosynthase domain sequence of polyketoid synthase gene from Aspergillus species, which is more specific than common ITS probes used for fungi. The detection methods identify aflatoxin producing and non-producing Aspergillus species from agricultural samples. The diagnostic assays use specific primers and fluorescent-based quantitative PCR assays to facilitate detection of A. fumigatus, which produces melanin, aflatoxin producing A. flavus, and A. niger from agricultural samples. And for specific detection and differentiation of Aspergillus fumigatus flavus and Aspergillus niger also in the clinical samples, which is highly relevant today for the corona patients. So, what three types of primers have been made by us? One set of primers will universally identify as presence of Aspergillus flavus, fumigatus, and A. niger in samples, whether it is clinical samples or agricultural samples. And then it is specific, is there here to show that many other things have been tested. And we have here the PCR showing that A. fumigatus, flavus, parasiticus, niger have given. For some reason, Nidulens did not give, Boraceae has given, Antareus has given, and many other things are non-fungal, non-Aspergillus things. So we did not have that one. And here is the common PCR, 450 base pair PCR, which recognize fumigatus, flavus, and niger. So relevant for identification. And here is a 600 base pair primer which we made, which will identify IA flavus isolates from isolated agricultural samples specifically. And then what we try to do is some of these isolates we have tried to take out see the screening of aflatoxin, say in these fungi. And we try to purify also Aspergillus aflatoxins. And in a couple of things we have purified, that is number 14, we have purified very lightly, it came. Others we have run the thin layer chromatography with the solvent extraction from the thing and purified B1 by immunoaffinity columns. We have purified that is based on the antibody based affinity column we have used to find out the aflatoxins in these strains. So during this one, what has happened, some of them have given the aflatoxin, some of them have not given the aflatoxin. So we consider that the things which are not produced and produced, these are the two categories of the Aspergillus isolates we could make that time. And then a specific probe to see whether the flavus detection with the producers and not producers of a 350 base pair primer, we have made that primer for that. And so all the sequences are done sequencing and they are deposited in the NCBI. And here the PKS amplification of producers as well as non-producers, and some of them have not got produced aflatoxin by PCR as well as by our experiments on the aflatoxin detection methods. I have not given the much detailed more slides on that one, but here the polyketide synthase amplification from ITCC Aspergillus isolates. Aflatoxin is produced by biosynthetic pathway. Anyway, case region of PKSA was selected, primers were designed, PCR conditions were standardized. DNA was isolated from flavors, all the three things, and PCR for PKSA region was performed for all of the Aspergillus isolates. A product of 380 base pair was amplified, which was sequenced for analysis of any change in nucleotide sequence. Here, in fact, I had not presented the data here, but what we got, and I discussed with the Dr. Dubey also at that time, some of them, upon sequencing the ketosynthase domain from the polyketide synthase, we found there are certain SNPs which are missing in the non-producing aflatoxin species. And when we looked into the literature, it is already known commercially there is an availability of a method of using atoxigenic isolates for the controlling of the toxigenic aflatoxin producing Aspergillus flavus. And already when I discussed with the maize specialist, Dr. Prasanna, and some others, they revealed that such a thing is already being used internationally by biofactories, which are at Kenya and Arizona. I think these are the places where the application of using atoxic isolates for the removal of the toxigenic isolates. This is a very novel method which is not used earlier, but may require a lot of studies to show they are not harmful to the humans by many other means. So now I come back to the questions of what Dr. Dubey has posed, what the problems are there, and whether we can think about it and we can talk about some of these things. How to remove aflatoxins from solid and liquid agri products? This is the first question. I do not know what the exact methods are all being used by the people now to remove the things, but there are many other possible things available to do it with the advancement of technologies of organic solvents, and then also the nanoparticles and also phytochemicals, which are saying that these are certain things which can be relevant for the things. But you can educate me about that and we can discuss on that, how feasible it is. Then the environmental control of Aspergillus spores and conidia is a very big question which I cannot really answer, and then everyone should be able to think something, and environmental scientists should think and do something. We have to see for that. And novel and affordable methods with high sensitivity and specificity for detection of Aspergilli in crops and agri-feed. What is there to do for this one? How do we do this sensitivity and specificity detection methods for the finding out the aflatoxins and then with the things? So here there is some literature which is up to the latest literature, which appears to say that there are nanoparticles which are developed of late in the last 10 years of the time, and these nanoparticles are being utilized for making sensors to develop, to estimate specifically, sensitively and quantitatively for the aflatoxins. And development of, not only that, development of selective, sensitive, simple methods for determination of aflatoxins, and simultaneously with the multiple aflatoxins, that different types of aflatoxins. Is there a method? Is there, as it is currently with what is available, and whether we can do something to go for such developmental for the diagnostics and detection methods is, I think it is highly relevant for that studies. And better understanding of host pathogen interactions for development of resistant varieties. Here, lot of genetic varieties need to be developed. I am sure that already the things are being worked out at CIMIT and several other countries connected to them. And these are the things where we require to understand basically host pathogen interactions, and then how the gene interactions take place, and what is there to manipulate, and how do you do these techniques for practical utilization. Then identification and characterization of atoxygenic flavus isolates, I feel, is a relevant one for the situation which is there in India, because in certain countries it is working very well. Cotton fields very well, it worked out, people have used it, and several other crops also people are trying to use it. But in our country, I have not seen much of work on the finding of these very useful and relevant isolates. So some area of this work can be done for identification for certain control of the crop contaminations. Then for this we require anyway that we require to characterize the toxigenic isolates and characterize them. When we try to deposit the SNPs, which we found in few of them, then we were advised by the NCPI and other organizations to carry out up to minimum 50 isolates to show the SNPs, then only they will get them deposited. That's where the challenge remained, and then we did not further continue on that part of the work. And the last one is a common challenge for all the scientists, because whether it is for the human or it is for the agriculture, all the compounds which are pharmaceutically relevant for human health, they are coming from the fungi and several other microbes. So because we have so much of literature available on the Aspergilli today, with so much of genome sequencing information, ESTs, and a number of biochemical pathways, and we know the initiation and termination things, and there is a good possibility to work on this also while working on the Aspergilli, to look for the biotechnological potential of these organisms for their highly relevant compounds. And then we can discuss on the points of last topic. If there is, I can help you in some way, that is there. And here I like to definitely acknowledge all my students who have been dedicated scientists working on Aspergilli and several other aspects, right, starting from Banani Banerjee, and Dr. Taruna is continuing greatly even today for the kit and also for the lung surfactant work. And Poonam is a proteomic expert. Wherever there is a need for proteomic work, I can definitely help people to get her help and can be directly approached. Shankar was on molecular biology, Shaili was on molecular biology, Savneet is now today in stem cells, but she is connected with the lung surfactant protein, and Lakshana did a great work on the lung surfactant protein and the cancer relevance. And Preeti is the last one bioinformatics work she has taken. I pushed her to do certain bioinformatic work, and then she's doing now in Harvard Medical School, working on the corona mutations and then those things. And Dr. Ruti Singh was a MD student. She is in the medical college, professor, doing a lot of research work. And Priyanka is in the industry. She is working on the vaccine part of it in Panacea, and currently she's in Gene. And Shweta is in DPAS. All of them are really very dedicated, and I'm very proud of my students. And also I will acknowledge all my collaborators and colleagues and all the people whom I interacted at IARI as well as at GIB, from all over the world. I'm very grateful to them. And especially IARI, I am very surprised because I used to stay at IARI and go to IGIB to Mall Road. But my last stage of thing, I joined IARI, continued to do the work, sometimes going to IARI still. Thank you very much. And I am ready to discuss anything and consult anything. Thank you.
Thank you very much, Madam, for very, very comprehensive and informative lecture delivered by you. I am hopeful that the purpose of the lecture will serve. This is the way, just 75 years of independence, this is the simple mediums to listen you. Otherwise, the purpose of your lecture is much more wider and much more important rather than the celebrating the 75 years of the independence. Definitely, I hope the organizers will get the certain points, especially for the diagnostics, very specific and sensitive diagnostics, molecular and serological based diagnostics, and maybe further can make a programs to refine them, how they can diagnose, and management of the diseases caused by these pathogens, especially for the Aspergillus flavus and other species of Aspergillus, because there are a lot of species. I remember from ground you analyze more than 61 isolates and about 36 or 26. Still, I remember that slide, 26 or 36 are Aspergilli producing. From different area of Gujarat and others, they have been collected. So definitely they will utilize your knowledge and make a very robust program for funding. I hope the purpose will be solved, and management aspects has to be addressed more in the projects. So although it was not widely covered in a lecture, because the area where you have worked extensively, you covered that one. So management aspect has also to be discussed with you later on, and they will devise the research strategy for which this can be done. Because once the contamination, as a pathologist, we all know, once contamination is established, it is very difficult to save or reduce the level of aflatoxins. So we have to avoid the contamination.
Yes.
And what would be the approach to avoid the contamination? Either any pesticide application or genetic manipulation by the conventional breeding programs to make the variety resistance. What mechanisms involves to association with the Aspergilli with the host, because this is very the facultative pathogens. This is the facultative saprophytes, and facultative saprophytes breeding a variety resistant against facultative pathogens very difficult, because they have a lot of mechanism of the pathogenicity. They have the enzymatic action, they have the toxins, they have the certain cases, they have the physical force also.
Yes.
So we have to find out the way by which genes by which we can make a plants more robust and tolerant to the Aspergilli. That would be best approach. And what else may be there? I think definitely it needs a lot of discussion among the group, and then accordingly the program has to be made in spite of beside this knowledge what.
Here, I would like to say that I have published a review article in which I have dealt with the management part of also to some great extent. It is in the 17. I will forward it to you. You can definitely look into that. And there are a lot of changes, and people have tried to do different, just not by drying, by using different compounds also, and then biologically also they try to control. And then a lot of things are there which are being tried.
Yeah, definitely a lot of approaches are available, but simply why we have suggested to discuss with you prior to formulating the projects, because you have the experience, and among these approaches which has the feasibility, which has the feasibility, not only for the point of the publication, but the technology which can be used by the producer and the farmers to save their crop, because a lot of consignment has to be rejected due to the high level of contamination of aflatoxins. When I was in the pathology or NBPGR, so many people are coming for detection of the aflatoxins in groundnut. So many people are coming for detection of aflatoxins flour, maize flour, even wheat flour. It is mandatory to certification is required. The flour is free from the aflatoxins. The quick and reliable method is required for detection, as well as to the methods by which we can avoid the contamination into the food after post-harvest process also. So these words, once again thanks, madam. I would see here Dr. A. Kumar is there. Welcoming you, Dr. Kumar.
Thank you, sir.
The organizers will get benefited from your lecture. Thank you very much for giving the very valuable and informative lectures. Now it is over to the IAMR.
Yeah, thank you.
Thank you, Dr. Dubey.
Dr. Dubey. And definitely I would like to help in whatever the way I can. I will go through also that, and I will discuss with the scientists and see that what best methods can be utilized for the control and control. I'll be more than happy to do that.
Other persons to whom you like to be associated with the projects, or other persons to whom the knowledge can be acquired, Director IMR will discuss, and accordingly, because we want a very robust project. So other piecemeal of work is not solving the purpose. And from last 20 years, a lot of groups are working in Aspergilli, and still the problem is remain as such.
Absolutely.
Substantially
at least this freedom after 75 years, we should come out of this problem to a great extent. Our products are economically and WTO stipulated situation.
I know this. Whatever basic information your group has generated, this has to be converted into technology and can be used by the producers and farmers. That's why. Thank you, madam. Thank you once again. And thanks to the organizers for providing me an opportunity to be here and listen.
No, I must thank you, Dubey, really. And I am so happy to see you also after such a long time. We used to have nice scientific discussions. I miss that in the plant pathology. You are not there. So, but now it's very nice to meet you, and wishing you all the best for all your efforts to see the betterment of tomorrow with all these agricultural crops. And economically, we should become much sounder than this.
Thank you. Thank you.
Thank you. Anybody can ask any question and we can discuss on that. Yes. Dr. Kumar.
Yeah. Thank you, Director IAMR, Dr. Dubey, Dr. Madam. It's a very nice, enlightening talk for me. I have a couple of observations and also our own perception about this aflatoxin, ma'am. You talked in length about PKS pathway and its role in aflatoxin biosynthesis. I have a very fundamental question, ma'am. What is the natural function of this aflatoxin from the point of view of Aspergillus?
That is the biggest problem. This is, see, secondary metabolic pathways in Aspergilli. Why they are there? Is it for its own protection, or it is trying to interact with the host to maintain its chameleon circumstances so that it can survive? Is a big question. We do not have still any understanding, clear understanding to say this is because of this it is having like that. There may be multiple roles of that.
Yes,
this is possible.
Yeah. Why I am asking this question? I happen to work in another project years back. That was on cyclic lipopeptides produced by Pseudomonas fluorescens isolated from some marine ecosystem. People worked out the natural function of the cyclic lipopeptide. And these cyclic lipopeptides are very powerful anti-oomyces compounds. People are trying to use it in oomyces control in several controlled greenhouses in Western world. When they address the natural function, what they found was they are actually protecting the bacteria from the predators, which is part of the microbiome where they are living. In particular, the protozoan predators. So that was a natural function. So I have a kind of strong feeling like aflatoxin producing strains must have some, they definitely benefit out of the secretion of this aflatoxin. It cannot be a kind of accidental release of this aflatoxin in the environment. This is one question, ma'am. Second question is, how relevant or vital the PKS pathway for aflatoxin for Aspergillus? Suppose if, can we shut down this pathway? Can the Aspergillus be still an Aspergillus leading its life in an ecosystem as a saprophyte?
It's a good question. So the point here is that the entire pathway is very difficult to be shut off. That is the main problem with the thing. Because the understanding on the entire pathway is still, it's a primitive role. We only got a certain amount of information till today to have the activation of PKS in something. They're like, what is it responsible? So terminators and initiators and regulatory genes, we have very little understanding. So that is one thing which complicates the situation. That's why I said we need to know the conditions under which these are triggered. And for example, very good example I give you, Aspergillus oryzae is there, right? You heard of that. It's a biotechnologically very useful. You use it for the food industry without any hesitation, what it is happening. But when we try to look into the sequence and comparison with the flavors, it has got polyketide synthase biosynthetic pathway, but it does not produce the aflatoxin. Then we realized it is cryptic there. It is cryptic there. So this is a great example to study the basic science to differentiate what is it triggering. What is it triggering the crypticness to the activation? That is one thing.
Yes.
Then physiological conditions and environmental conditions of the growth of the fungus can give us a certain information if a basic study is done with respect to the environmental conditions plus the secretions of the flavors and the synthesis of the compounds, what it is making. That can also give us an insight very simply, but no studies have been done to do these things in a methodical manner to get the outcome, to correlate it properly.
Yes, ma'am. Madam, in connection to that only, you talked about it is typically an inducible system in Aspergillus, the PKS, or it is constitutively produced. If it is inducible, then it must get some cues from environment, in particular the abiotic environment. My question is there any record of aflatoxin biosynthesis in the pre-harvested produce? That means when the cob or in the groundnut is still with the plant, it is not harvested and gone into the post-harvest storage. The pre-harvested produce, is there any aflatoxin secretion? Or it is accelerated once it is detached from the main host, then it moves to the storage. Then the PKS pathways are activated. Is there any link?
No. I don't think it is doing that. It is doing it while it is a pathogen on the plant itself. The reason is very simple. When plant produce, you isolate the flavors, it is having the aflatoxin. So that secretion should be happening when it is already as a while a pathogen.
Okay, you mean to say that in pre-harvest
It is not
produce
just
also it must be there, yeah.
Yeah,
Okay.
but the quantity can be different. That is possible. When
yeah.
flavors contamination is there on the so much of feed, it has got so much to produce, so it multiplies and it does. But when it is on the plant, it has competition with many other microbes too, and it has to survive on that.
Yeah, that is what precisely my question, ma'am. See, the threshold level, which is creating problem in the marketing of the produce, we don't get that level in the pre-harvest storage, pre-harvest material, but we get the post-harvest only we get this problem. So that is what the question. The PKS activation, what is the trigger for this activation? This is a density dependent kind of quorum sensing is operating in Aspergillus, or you rightly told when the pre-harvest material, it has to compete with so many other natural microflora, what I call microbiomes, and it is under the check of those microbiome or their metabolites, so that it doesn't really secrete Aspergillus aflatoxin in a quantity that is detrimental for the produce. So my question is, what is the signal? It is the harvesting from the produce is a signal, or it is a kind of environmental condition where it is getting stored it's a signal. I think this is something we need to really address.
Yes.
What is your comment on that, ma'am?
Yeah, the studies have to be carried out there. See, when you are looking at the product, that are you looking at only aflatoxin, or are you looking at any possible existence of A flavors there? Are there methods to identify them when the produce is there, both for aflatoxin as well as for the flavors? Because it can survive under different variable conditions in a manner which it suits it. So, are we doing this routinely now in the products, that when you make agri-feed, for example, for animals, the agri-feed is looked into for only aflatoxin content, or are they looking for the presence of the flavors?
Okay. Yes, ma'am. Just for your own information, we had a project which it is still going on, where we are addressing the cob microbiome in maize. We collected the material from 2 different agroecological zone, and we analyzed the cob microbiome. What we found was within the microbiome also we addressed the microbiome, the fungal diversity. The surprising information was the cob itself is having rich diversity of fungi. That was not there in the rhizosphere, number 1. Number 2, among the fungi, what we encountered was it's all Aspergillus, variety of Aspergillus. You name one, we found Aspergillus fumigatus, flavors, Niger, and I think there are so many others. I'm not recalling them. But the point here is we really do not know whether they are toxigenic Aspergillus or they are normal microflora of cob. That we really don't know. So that is one story. So another story is the same cob, what we found at some unique microbiomes, the prokaryotes also. So we have a thinking like, if you can manipulate the microbiomes on the cob, there may be a possibility to remove this toxigenic Aspergillus from the cob itself right during the harvesting time. So this is something like what other people are using, the non-toxigenic strain. And we prefer to go for other microflora rather than the non-toxigenic strain, because Dr. Dubey very rightly pointed out that toxigenic, non-toxigenic, it is a very slender difference. Actually, anything can happen to the non-toxigenic also. So we would prefer to go for the native, the beneficial cob-associated microbiome. Some kind of manipulations there by number or kind of activation of some metabolic pathway. My one question is there any inhibitor of PKS pathway, ma'am?
I don't know, that of late I have seen that some phytochemicals, when they are treated with the Aspergilli, then they are not able to sporulate, and then there is a difficulty for them to synthesize after. But this, how much scientifically we are able to demonstrate with molecular level, I do not know. But then there is a paper which I recently saw that they are using a phytochemical, that name also, I can forward that to you, very last year's or before last year's paper. It's a phytochemical, they are using that for the reduction of production of aflatoxin. So if it is really doing it, then that sort of a compounds are highly relevant for us. Such sort of a compound treatment right from the beginning, which is not harmful. A phytochemical is not expected to be very harmful to a treatment too. So it's not a organic chemical you're synthesizing and using. That may be of relevance. And such type of work is also going on in some parts, even in India. I think that some group was trying with oils and some group with this compound. I will to know
so
that.
that means it's a matter of deep exploration of the cob itself.
Yes.
To find something which is produced by other microbiomes so that they interfere in the PKS pathway. Anyway, I'll stop it here. I think there are so other scholarly people are sitting here. I think they can interfere and they can come out with some wonderful suggestions.
Yeah.
Thank you. Thank you.
Yeah, nice talking to you.
Interaction.
And then I too enjoyed learning from you that you have got so many Aspergilli there. My God, that is there. See, we have not identified many other toxins. We are talking always about aflatoxin, but not aflatoxin alone. There are many other toxic compounds produced by these organisms. So the best thing is to remove that. Try to get rid of the organisms as early as possible.
Yes, from the I think director is already here. I think we can make some.
Thank you, Kumar. Nice talking.
Yeah, we'll keep on interacting.
Yeah, sure, absolutely.
Thank you, madam. So now I request.
Yeah. I can't hear.
And there is some voice break.
Okay.
Yeah, there is a voice disturbance.
Yeah,
I am actually located at such a place. There is some problem in network.
Yeah.
Oh, I see.
So, anybody else wants to ask any question?
Yes. Good morning, ma'am. I'm Ala Singh from Indian Institute of Male Research. Ma'am, while hearing your talk, I was browsing sidewise, and I found that because there was mention of Aspergilli-Advances, which is a non-hazardous organism, so to speak, but there have been cases of hypersensitive pneumonitis in koji breeding because of this Aspergilli-Advances. So, ma'am, based on the pathway that you showed, that surface proteins can cause phagocytosis.
Yes.
So, ma'am, again, the question that now appears is that can non-toxigenic Aspergillus cause lung infection in at least immunocompromised individuals? Because this pathway would kill us.
Yeah, I got your question. See, basically, Aspergilli, take them, all Aspergilli are human enemies because the spores are there and the conidia are there in the air, and they have proteins, proteases, and elastases, which are all, most of them are allergic to humans. Take it that way. Because of the environmental conditions, you find A. fumigatus as a dominating fungus at one point of time. In 90s, I started fumigatus. Why I started fumigator? Because fumigatus was the major concern for the people based on the skin allergenicity test. And later on, flavors has been reported. Niger has been reported. Even Teresa has been reported. Oryzae is reported. I'm not surprised. So all the proteins of these fungal spores, especially spore for having, which are there in the aero, you consider them as aero allergens, unless you tested positive, negative, that you are not negative. But all of us don't develop the disease. That is the important point, that it depends on the load of the spores. For example, in the latest corona situation, and there was oxygen concentrators with a lot of tubes, people. And then you have seen in the papers that when people have gone to the hospitals, came back from the things, they developed mucormycosis and mycosis disease. That from where it is coming, it is all coming from the body does not produce aspergillus. As it takes, it multiplies, it becomes invasion for that. So no surprise of Oryzae or Niger, Parasiticus, any of these things, depending on the situations, their compounds are allergenic. We have not identified which components of these are allergenic. Very few things have been done. It's an area of, again, lot of concern for human health, but no characterization, not much detailed studies have been carried out.
And then this calls into caution the use of various competitive exclusion strategies using non-toxigenic strains to control toxigenic Aspergilli. So then it needs further investigation for that as well.
See, that is why I clearly told, when you want to consider a non-toxic, right now people have done these isolates, non-toxigenic isolates controlling the toxigenic Aspergilli in many places. Kotti was the person who has done a lot of work. I was seeing his paper. I requested him to send the isolate. So I wanted to compare the gene sequence of my isolates which are not producing, but I could not get it. There are so many agreements and procedural problems. I couldn't get a comparison. So question is, that's why when you want to declare this is something for the field work, for any type of field work, from the Aspergillus group of the things, one has to be cautious about what is it producing. Okay, toxigenic, that is ठीक बात है. But then if it is producing a lot of allergens, then what are you doing? Are you doing a good job here and then making the public exposed to the sports? That is not right. So there is a clear-cut demarcation of knowing. And how do you do this test? You can ask this also to me. I can answer you. If you find an isolate which is non-toxigenic and which is having a vegetative propagative help, it definitely can be used. Now, how do you say it is not allergenic? We have allergic models. We can develop animal models. We have developed. That's why the earlier work I showed is how a compound can go from one place to the other utility by doing so much of sequence of science and still coming to a viable commercial product. What we have to do, we have to develop animal models. And we have to use those animal models for the effect of these allergens. We can take out the allergens, protein fractionated proteins and purified proteins out of these culture filtrates and mycelial. Science is now advanced. You have every technique to do it. And even a attomoles protein molecule is enough for you to do so many great things. So feasibility is there. Technology is there, whether it is for human or agriculture. How you apply it for your problem, your question is in your hand. I strongly feel that.
Okay. Thank you, ma'am. Thanks a lot.
Thank you. Thank you very much for. Yeah. Anyone else? Yeah. I can't hear. I am not able to hear. Voice is breaking. Voice is breaking. Yeah.
I think still voice is breaking. I think a lot of disturbance.
Okay.
Yeah.
Dr. Usha, can you hear?
Dr. Rao is there, right? Nice to see Dr. Rao.
Can I ask a fundamental doubt actually?
Please ask Professor Rao. You have been very helpful to me for peptide work. I just acknowledge that.
Since it affects the lungs, does it have any receptors? Like you compared with COVID, where it has the ACE2 receptor, like your sports, does it have any receptor on the lungs where it causes the...
You are talking about the lung surfactant protein? Yeah, okay. Lung surfactant protein is competing with the coronavirus for the ACE2 receptors.
Oh, okay.
In a dose-dependent manner. This is a recent publication, 2031. And Karuna also has done a lot of work where they showed this lung surfactant is helpful in controlling the infection and multiplication of the virus. Which is a very strong point for approaching the lung surfactant protein to be a considered a molecule for the as a nasal spray or something like that, because its innate immunity makes a strong, powerful candidate. And how it is doing is proved now. And then the evidence is there. So that is where the lung surfactant is taking a good shape now to make whether for an inhaler, and Taruna is doing all that work in collaborations, and they have a BIRAC project where they are going to go for the GMP controlled product preparation and testing in various hospitals and things. That is the rest of the, all credit goes to, only I wanted to show that how the lung surfactant work which we have carried out, basically relevant for the diagnostics for today also, not only for Aspergillus, tuberculosis, it's relevant for the mycosis in the corona patients.
Correct.
And therapeutically, lung surfactant is highly relevant.
Hmm.
And then how basic science can be used for the practical utility for the society in making it a viable commercial product. Even for aflatoxin testing, we submitted a patent for the detection methods. These methods can be taken forward to make into a more simpler and easy manageable methodologies. That can be done. Basic science, and then what we found is we have given. Now it is up to people to utilize it, how they can be done. Yes, Kumar.
One
more from
Dr. Rao is saying some more questions.
Some clarification, only one clarification.
Yeah, no problem,
Okay, sir.
Please.
Okay, sir.
No problem.
Since it is a natural molecule which is produced within the body, have they tried in the corona patients the lung surfactants as a IV through IV pad, something like that? Have they tried actually? Because there's no harm actually. It is a your own body producing different types of lung surfactant.
Yes, here I like to tell you, this surfactant present in our bronchoalveolar lavage as well as it is present in the type 2 Clara cells of the lung. Okay.
Correct.
And this is the full length protein. It's a very huge protein, and then complex. I already explained to you. So what we try to do is we developed with the, actually it is Dr. Uday Kishore and KBM Reed who developed the recombinant form of the lung surfactant, especially D. So whatever the experiments are going on for the testing is with the recombinant form of the lung surfactant D. Not only that, they showed it is a better one than the whole molecule. Basic reason is you can't purify from the, how much bronchoalveolar lavage is not. So the recombinant form is now going for the application part of it, because enough science has been done by us and several groups also in some other bacterial viral things. So it is a very potential molecule. It's a innate immune molecule. So you can you just have the how it gives the strength by this one.
Okay, thank you.
Are you satisfied, Dr. Rao?
Yes. Because for corona, there is an animal model already developed.
Yes.
Corona animal model already developed. Hamster model is there. They can do in hamster model and see how the recombinant the lung surfactant is really beneficial to the host. And
also Taruna Savneet at ILBS in Delhi, they are developing now animal models to test that surfactant, recombinant surfactant.
Okay.
So
no, already people have shown the hamster is one of the model they showed.
And already it is there.
Okay. Thank you.
Thank you for educating me also about the hamster.
Okay,
right. Kumar has got some more.
Yes, madam. One offshoot doubt, it's an ignorance only. This aflatoxin producing strain, the Aspergillus flavor, whatever you call,
flavors.
Are they produce any volatile compound that is very toxic to human? You come across with any literature? In addition, I believe aflatoxin is a secreted metabolite.
Yes.
But what about
they are coming in the they are coming in the culture filtrates. Yes.
Exactly, ma'am. So whether the aflatoxin has got the tendency of getting released by Aspergillus in the form of volatile?
Volatile, I have not heard and I have not come to know. It will be a good knowledge for me if I look into that part.
Okay. And that's just curiosity. I just wanted to ask.
It's a good question.
Anybody else?
Excuse me, ma'am.
Yeah.
Namaste, ma'am. I'm Sriniketan from ICAR-IARI. I'm a PhD student.
Okay.
Ma'am, I am working on Aspergillus, which is used in bioremediation of contaminated soil. So, if I may ask, would you suggest some basic assays for toxigenicity tests apart from kits? Because I've been doing some plate assays which prove that these strains do not produce aflatoxin. So, are there any other tests which are mainly related to agriculture?
Other tests for the aflatoxins, you mean?
Yes, ma'am. Aflatoxin producing.
Aflatoxins, they say that we did the thin layer chromatography, which was there as and then that is only qualitative, right? And then maybe there are densitometers which can be used for the quantitation, with taking out the spots what you have. Besides that, ELISA methods are the ones which are there, and you can develop a spot methodology based on the ELISA technique. a dot blot type of a technology can be developed. That is a possibility because simple. A dot blot is much simpler than going for all the things. So the dot blots are available for the human serum samples and human testing for many of those things, you know, even like a pregnancy test so that you see the thing positive, negative. But here you need a quantitation. So depending upon the diameter of the dot, you can measure the quantitation also. So simplification. Here what happens, Shiva Ketan, that there is science available. There is a lot of science for estimating, detecting, finding out things is not one method. Molecular methods are there. And as I told you, we also put a patent for that recently for simultaneous detection of flavors, fumigators, Niger, and differential determination of these things and specific detection of producers and non-producers. These are the things we could, what we got out of ketosynthase studies is these are the things we could get. We could understand certain things can be possible for doing. Now, simplification of this is the younger. You people have to go for a little more dynamically to see what are the things which are available, whether you can use that technology here, right? Because technology is available. Whether it is for human or agriculture, how does it matter? Your question is the matter. You question. What is the question is your original thinking and question. That should be answered by any means, right?
Yes,
so
yes,
I feel that possible to be quantitated. And what we did is we did the ELISA. Yes, we did the ELISA. We did the TLC, identified. And then we also try to do the densitometric comparison, which is a semi-quantitative. Densitometry is a semi-quantitative. It doesn't give you the perfect quantitation, right? So that is what we try to do that.
Can the kits developed by you be used for agriculture purposes as well?
The kits developed by me is for the human detection of antibodies to Aspergilli. It is in the serum based, right?
I'm sorry, ma'am. I'm sorry.
But here it is not. But the patent which we have submitted is relevant to you. That molecular probes which are there, RT-PCR. You can make an RT-PCR, quantitative RT-PCR. You take it forward and make an quantitative RT-PCR, though it is a complex test than simply looking at the thing, but then that will show you even the numbers of the spores you have got, but that is for detection of Aspergilli, not for the aflatoxin. What we
Thank
have
you so much, ma'am.
patented that one.
Thank you so much.
Thank you. Anytime you want to talk or discuss with me, I'll be more than happy. I always
Thank you so much.
students to
Thank you.
interact with that. Yeah. Thank you. Anything else?
No, thank you very much, madam. I think Dr. Sujay is not on line.
Okay.
Now I request our colleague, Dr. Dhanpal Chaudhary, to propose vote of thanks.
Okay. Thank you very much, Sekar, for your.
Hello, am I audible, sir? Sir, am I audible?
Yeah, you are there.
Yeah.
So before I say vote of thanks, I request all participants to please switch on their video. We want to retake the snapshot again. So it's my pleasure, madam, to say vote of thanks to you who gracefully accepted our request to deliver a special lecture on this Aspergilli-Advances and challenges. So it's a very nice talk, madam, very beneficial to us. You have very nicely highlighted the importance of protein interactions in Aspergilli infection. Then the use of recombinant surface proteins in therapeutic management is very interesting. Then, madam, I noticed few points: the elucidation of genetic pathway and the intricacies involved. You explained that will be, I think, very helpful for future research. And finally, ma'am, you very nicely highlighted the challenges for future research. That's very interesting.
I can't hear you.
Dhanpal, you last. You are audio.
Mute. Dhanpal, mute. You are muted.
Okay. Sometimes I think I don't know how I'm muted.
Sometimes
finally,
it happens.
Okay, ma'am. So finally, you very nicely highlighted the challenges for future search. That's very interesting. So I'm hopeful that the entire Mage family will get benefited from your talk. And I also would like to say, would like to thank Dr. S.C. Dubey, our ADG PP, and Dr. B.M. Prasanna and other my colleagues from ICAR, from ECRIP centers, as well as from IMR. Thank you all. And finally, I would also like to thank our esteemed rector, Dr. Sudhir Rakshit, who connected with you to deliver this special lecture. Once again, thanks to madam.
Thank you very much for all of your listening, and I am sure that anything you want, I am ready to help you. And it gave me good opportunity to be sharing some of these things which are relevant for our health as well as for agriculture. And I am open for any type of interactions we would like to have. I will be happy to do that. And wishing you all the best for great achievements. Thank you very much.
Thank you, ma'am.
Thank you.
Thank you, ma'am.